Okay, so today's class is on genome assembly. Obviously, a very basic class because it will be covered in much more detail in a course called genome and proteome, and sorry, genomics and proteomics. But still giving you an idea that once you get the sequence of a particular organism, then what are the various approaches? How do you assemble the sequences? So in our last class, I told you that what are the various sequencing strategies. So for simple sequencing, you just go for Sanger sequencing. Even now it is being practiced, automated Sanger sequencing. And then I also told you about the first generation sequencing method, which is pyro sequencing, the parallel pyro sequencing method. And now, of course, there are so many second and third generation sequencing methods that have come up. And if you just go to, even Google or Wikipedia, you'll get so many different strategies, solid sequencing and ion torrent and nanopore. And there's so many, means it's such a dynamic field, at least with respect to sequencing methods. Obviously every sequencing method you'll find it as interesting as probably pyro sequencing, because even pyro sequencing I thought it was very very sophisticated in nature, but now new sequencing methods have come up. But in all these sequencing methods, of course, the assembly is one issue, and how do we address, and which are the various methods by which you go for that, is today's class. So obviously we have so many different organisms which differ hugely in the genome size, and you can see like 0.58 MB to 16 MB, etc., KB, etc. So 16,000 MB, sorry. So means obviously if we have to sequence something very small in size, let's say any plasmid in current times, or very small organism, virus in current times, then obviously even Sanger sequencing will suffice. At one time, Sanger sequencing generates 700 around base pair as good reads for the same. So if you have a plasmid of 4 kb, so you can go step by step, you sequence it from both sides. So at one time 1.4 kb will be covered, then whatever the sequence comes, you generate a probe or a primer from the ends of it, and then again sequence further 1.4 kb. So that way, if you go step by step, even for very small plasmids, etc., you can easily go for Sanger sequencing. But the issue comes when you have very, very large genomes, let's very means mammals and plants, etc. So in those cases, you have to resort to these parallel sequencing approach. And which approach we go for, I just come to that. This just gives it's like a straight takeaway from T.A. Brown book. It's a snapshot. And you can see that just for your information, that the first DNA molecule to be completely sequenced was around 5 kb. It was a bacteriophage. Then again, it was around 5 kb. Among the virus, it was this one. Then PBR 322, almost 3 years later. Then mitochondrial genome 16.6 kb by Professor Sanger's group, and bacteriophage lambda 49 kb, if you remember, in 1982, etc. Okay. So among the sequencing methods, obviously it will be parallel sequencing. But which are the two methods of genome assembly? So there is one shotgun approach, and one there is clone contig approach. So in shotgun approach, it is based on the fact that you break the genome into small size, and you generate a library, and you sequence each clone separately, right? So all these clones, they are sequenced parallelly. They are sequenced parallelly, and then you look for the overlaps between these sequences, and then you generate the whole genome sequence. Okay, so basically you take any organism's total DNA, you break it into small pieces, you do parallel sequencing by any of the methods that I have not covered, and there are so many different current methods. And then you look for the overlaps between these sequences by, so you feed these sequences on computer, it looks for the overlaps, and that's how it assembles the whole genome. Okay, so sorry, this is the one, this is a shotgun approach. Then another approach is that you generate you again means similar, that you break the genome into small sequences, you generate the clones for each of these, and as a first step, rather than directly going for sequencing, you look for the these overlapping clones first. So what you do is, in a way, you pre-assemble them in the form of small contigs or continuous sequences which are overlapping with each other. So let's say if there are 100,000 different clones here, then in this, let's say we have assembled them into 20,000 different clones. How? By already generating a series of overlapping clones. So once you get one whole contig in place, and then you sequence that particular contig separately. So what is the idea behind this is that when you have the whole sequence, when I'm talking in terms of, let's say, human or plant, where the whole the size of the genome is very big, in those cases assembling becomes a very big problem. So it is considered better that how about we have already a set of overlapping clones, and how we generate those overlapping clones, I'll just tell you, there are various approaches for the same. And once we generate one contig, then we sequence that particular contig, and then means that sequencing can still be through shotgun approach. In a way, you have divided your whole genome into small contigs, and then you sequence it. Is it clear or not? Am I audible or not?
Yes, ma'am.
Yes,
You are
ma'am.
audible.
Audible.
So, did you get the advantage of clone contig over shotgun? इसमें बहुत सारे स्मॉल सीक्वेंसेस होंगे, उनको एंड में ओवरलैप करना विल बिकम वेरी वेरी डिफिकल्ट. एंड व्हाई इट विल बिकम डिफिकल्ट, आई कम टू दैट. मेनली इट्स बिकॉज ऑफ द रेपिटेटिव सीक्वेंसेस. बट इन केस ऑफ क्लोन कॉन्टिग, आप पहले ही उनके ओवरलैपिंग क्लोन्स बना रहे हो, and then you are sequencing it, so assembling it will be comparatively easier. Okay, so now coming to this, the first, this Haemophilus influenzae, how its genome was sequenced. So you sonicate. So there are various methods of breaking the genome into small sizes. You can use enzymes for base cutters with overlapping fragments, or you use sonication. So in general, sonication is preferred over enzymes because it will be more random in nature as compared to when you use enzymes. So once you generate these fragments, then you run it on gel. And why you need to run it on gel, and why you have to optimize the method of sonication, or even if you're using enzymes, is because you need to clone them into a particular vector, and each vector has its particular cloning capacity. So if I'm using a back vector and I sonicate my DNA fragments in such a way that they are just, let's say, 10 or 20 kb fragments, then there is no use, right? So I have to optimize my sonication or through enzyme digestion of DNA in such a way that I get DNA fragments in a particular size range. So that optimization is always done before, depending on what size of DNA fragments you are going for. So in this case, I think they were going for means it's a very means among the first experiments that was done. So in this case, the DNA fragments were just 1.6 to 2 kb. But obviously, means if you're using some newer vectors, you will use a much bigger DNA size fragments here. Okay, then you will take these fragments, and then you can add adapters on either end, and then you can clone it into the vector of your choice. And then you sequence each fragment separately. Either you have already cloned it into the vectors and generate a genomic library, or if you are not generating a genomic library in the form of clones, you can also, you don't need to clone it. I mean, you can do parallel sequencing and then just assemble the genome. Anyway, so here the clone library was made, and then the ends were, the overlapping fragments were looked for using computer, and then the whole genome was made, means it was assembled for this particular organism. Now, the problem was that always you don't find that you get overlapping fragments. There are many cases that, let's say, this whole is the genomic sequence. You get the sequence through overlapping fragments still here. Okay, and you get another fragment till here, but you do see a gap here because you don't see any overlaps there. So then how do you fill the gaps is one problem. And what strategies are used, I'll just come to that. So in this case, what they have done, let's say you had three different contexts, and how these contexts have been made by putting together these overlapping fragments. Okay, now I want to find the overlaps between the same. So what I do is I do the end sequencing. I know the sequences of the ends, of course. So I generate probes for these, the oligonucleotide probes from either ends. And then I hybridize the whole genomic library using one of these probes. So let's say I start with probe number 2, which is from the end of conteg 1. So when I do the genomic library in this multi-well format, I find that, okay, my clone is hybridizing to this particular clone. Okay. And my this probe number 2 is hybridizing to this particular clone. And there is a possibility that I see that this probe 2, it hybridizes to 3 different clones. So then I sequence, it means that these 3 different clones, let's say there were 3 different red spots here, they all might be overlapping. So then by sequencing those, I can figure out that, okay, these 3 clones are continuous to each other. Okay. Now, another possibility is that I probe it with oligonucleotide 2, and I probe with oligonucleotide 5. Then, incidentally, I find that they both hybridize to the same clone. So it means that my conteg 1 and conteg 3, they definitely are in close proximity to each other, and they might be overlapping. So then in the, when I'm assembling the genome, I will put conteg 1 and conteg 3 as continuous. So through hybridization and looking for overlapping clones, through hybridization, I can find, or let's say I can finally fill the gaps. So this is how this organism was finally sequenced. And the strategy to look for, or let's say filling the gaps, is by looking for the overlapping clones. Now, one problem that is often seen with shotgun sequencing is the problem of when there are these repeated sequences. So this doesn't pose any problem for those organisms or for those plasmids or whatever what we are going for, in which the repeat sequences are not much. It is, if the repeat sequences are not much, then shotgun approach is a good approach. And especially for smaller organisms, we still prefer shotgun sequencing because it is faster. But those organisms where the genome has lots of repeated sequences, all humans, plants, etc., where it is full with repeated sequences, it becomes a problem. And one example is given here. So you have, let's say this is the original sequence, and you get 2 different clones in the sequence of which is this. Okay, sequence 1 and sequence 2. Now, let's say obviously I don't know the upper portion at all because I have to assemble it. what I get is these two sequences, and I find, okay, wow, sequence 1 is overlapping with sequence 2. It means the final sequence is like this, which is definitely not the case, because there is a middle region in between. So because of repeats many times, I can totally assemble my genome in the wrong way. And in this example, the identical repeated sequences are shown in the same DNA molecule. There is a high probability that, let's say, sequence 1 is coming from chromosome 1 and sequence 2 is coming from chromosome 10. So I won't know that. So what I will do is that I will assemble these two together. So this becomes a very, very big problem, and I can do totally erroneous genome assembly. So in those cases, definitely it is preferred that you go for clone contig approach. At least I will know that these means what I'll do, that I will generate these, let's say it's a big fragment is this, I will generate these overlapping clones here. And once I know the overlapping clones, then I will go for the sequencing. So what will happen, that at least I will know that these two fragments are in close proximity with each other or not. That I will come to know through my overlapping clones. So coming to this clone contig approach, you can see that it does not suffer from the limitations of shotgun sequencing, and more accurate sequencing is sequence or genome assembly can be promised with the help of this particular approach. And how do you generate these overlapping sequences is one point, and there are various ways by which you can do that. So means my final thing is that when I generate this my genomic library is represented in different clones, right? Now somehow I have to bring these clones together which are overlapping with each other without sequencing them. So because if I sequence them, then obviously it will be shotgun approach. So first I have to bring these sequences together. So how do I do that? There are multiple approaches. I either go for chromosome walking or IRE PCR or SDS assembly. And let's discuss these one by one. So chromosome walking. So in this, what I do is I randomly take any one clone. Okay. I generate a probe against that, which is done with the help of, you can add radioactive nucleotides while doing replication or transcription. And what I will generate is a DNA probe, which is complementary to this particular clone. Let's say clone A1 I start with. Then I do the library screening using this probe. So what I will get will be some overlapping clones, of course, because obviously there'll be some clones which will be having the same sequence like A1, at least part of it. So then I find that I finally come to the fact that, okay, A1 probe is overlapping with B4 and I4. Okay. So it means that these three clones are in close proximity with each other. So at least I have something to get started with. Then what I do is that I generate probe from the ends of these assembly that I've made. So I generate probe for I4. Now I find that, okay, I4, it's not only hybridizing with clone A1, but it also shows hybridization with F2. So now I know that B4, A1, I4, and F2, they are close to each other. So this is how, step by step, I walk the chromosome. Okay. I randomly start with any particular chromosome with nothing in mind, and then I try to build up the whole sequence of the chromosome. So this is called as chromosome walking approach. But as you can understand, it will be very slow in nature, because one by one, you'll have to do the screening. Screening is not an easy process. You have to do southern hybridize, you have to generate probes, and then you have to see that, okay, these are overlapping. Then you have to generate probe for the last one clone that you know. Then you again, walk further and so on. So it's a slow approach, but yes, for smaller, assemblies, etc., still chromosome walking is one approach that you can go for. But obviously, for much bigger genomes, it is not a good approach to go for. Means it will take time, basically. So we need more rapid approaches, and in this context, I'll introduce you to some other methods. And the whole idea is that you have to find overlapping clones without knowing the sequence initially. Okay. So one approach, one idea came that, okay, let's say this light blue clone is matching with this, let's say, green clone. Then the restriction fragment map of this clone should have at least some similarity with this clone. I'm not saying that these two are identical, but they are overlapping. So there is a possibility that one restriction, the restriction enzyme sites which are present here, they also will be present here at least for a part of the whole clone. So if they both are showing same restriction fragment pattern, it means that these two are overlapping. So what they do is that they cut these clones with certain restriction enzymes, then they run it on gel, and then they look for possibility of overlapping, or let's say same restriction enzyme fragments that are generated. In a way, it is also called as RFLP, restriction fragment length polymorphism, also called as clone fingerprinting here. So the simplest approach is to digest each clone with one or more restriction endonucleases, and to look for pairs of clones that share the same restriction fragment size. And obviously means the vector also will be generating certain restriction fragments in every case, and you will obviously, not give any weightage to the same. I mean, you will not consider those vector fragments. So that's how you finally build the whole assembly. And so it is not step by step, unlike in case of chromosome walking. Here you can simultaneously look, means you just cut these various clones with restriction enzymes, you run it on gel, and at same time you can find maybe this is overlapping with this is overlapping with this, and this, and this, and this is overlapping with these three, and so on. So simultaneously, overlapping clones can be generated from various areas in the whole genome. So one problem obviously could be that there is a possibility that two different sequences, they generate exactly same size of the restriction fragment, even though the sequence was different. It is like, it can be a chance event that exactly same restriction enzyme sites, they are far, means two BAMH1 sites are far placed, 5 kb in one case, another case also 5 kb, but the middle region and the whole clone sequence is actually different. So in those cases, you can do some error in overlapping. Another method is, again coming to the same figure, is that as I told you, that restriction enzyme sites can be common in the overlapping fragments. Similarly, when we talk about these repeat elements, so repeat elements in these big genomes, they are very, very common. And obviously, there is the possibility that the overlapping fragments, they let's say in this case, these repeat elements are placed 5 kb apart, right? So if this is overlapping with this, so here also it will be 5 kb apart, but in rest of the things it will be, let's say, 1 kb apart or 100 kb apart, and so on. So it means that if I do PCR using the primers which are specific for that repeat, then I will get a 5 kb fragment here also and here also. Okay, so that approach is called as interspersed repeat element PCR. Okay, so these are the two identical repeats. You generate primers against the same. And how will you generate primers without knowing the sequence of the whole genome? Is because certain repeats, they are common across various species and all. You can just generate any random, means these primers against those repeats which are already known. So what you will get is a particular fragment, not fragment, amplicon will be the right word. And when you run these PCR amplified fragments on gel from this particular figure, oops, is this visible?
Yes, ma'am, it is visible.
Okay, okay. So yeah, so from this particular figure, I can figure out that okay, clone 2 and clone 3, they do have this same size band, which has come from the amplification from these repeat region, and likely clone 2 and 3, they are overlap. Okay, so again, in all these methods, there is a chance that you can do wrong overlaps, but then you can always cross check it using other strategy, but definitely it will give you a starting point that likely these two among several millions of clones that are generated, at least you can shortlist that okay, likely these 5, 10, they are overlapping amongst each other, and you can cross check the results that you get from IRE PCR using maybe restriction enzyme method. Okay, so this is another method of also means again it is rapid as compared to chromosome walking. And third method is STS based mapping, in which it relies on the fact that one sequence, this is sequence tagged sites, which refers to those genes or sequences which are present only once in the whole genome. Okay, so it means that if I generate a probe against that particular site, then I can figure out that if clone 4 and clone contact 2, these two contacts, they are, these two clones, they both show hybridization to a particular STS. It means that 2 and 4 are definitely overlapping with each other. Why I am saying definitely? Because I am very sure that this gene or this particular sequence is present only once in the whole genome. So if two different clones are showing hybridization with the same, it means that definitely these two are overlapping. So even this method can be used, but in this case, you need to have some prior knowledge about some genes which are present in that organism already. How will you know the knowledge of certain genes which are present in that organism? By some, some studies which are done by scientists who are not as such involved in studying the genome sequence of the organism, but for various purposes they are interested in that particular bacteria or yeast or a particular mammal species, that they're working on a particular gene. And when they do this, that in how many copy numbers this particular region is present, then they come to know okay, it's present in only one copy. So then they say that okay, this is sequence is one which is present in only one copy, and that can be considered as a STS, sequence start site. Now, so I've told you that to look for overlapping sequences and then build a contact. So let's say this overlaps with this, and this. So what I do is that these four I put together, and then I say that it is one contact, one very big contact that has been generated. So now, once I put these four contacts together, and what I will do that I will now break this very big contact in means it can be few MBs also, into small pieces by shotgun, means by breaking the genome, and I go for shotgun sequences. Now assembling these four the sequences reads which are generated from these four together, the assembling will be much easier as compared to अगर मैं इन सबको एक साथ छोटा means I break it into small pieces and then I sequence it, because then I'll be confused कि okay, मेरा जो repeat यहाँ आ रहा है हो सकता है वो यहाँ पे भी आ रहा है, यहाँ पे भी आ रहा है, यहाँ पे भी आ रहा है. So my assembly will be much difficult as compared to if I am just restricted to these four fragments, and I know that whatever sequences are being generated are definitely as part of overlapping clones, and I can put these together. Now, are there some other methods which can help me in assembling the genomes? Yes, there are various genetic maps which have been generated by various scientists or geneticists who have been working on that particular organism, by which they have placed certain genes together in the genetic maps. How they have put it together? With the help of, see, you have to remember that Mendel's experiments and all. So he would see that, okay, these two traits, are they always, what is their distribution in the progeny? So if these two traits, they always stay together, then they are placed close in the genetic map. And if we see their segregation, percentage or segregation frequency, that they are never found together, then they are placed far in the genetic map. So these are means, I hope that you remember that these experiments Mendel had done, in which he was looking for certain traits and he wanted to check that how often they stay together, or they are differentiated and so on, and why will they be separated. So if, let's say, these two traits are far from each other, then you will never find them together in the progeny, because these through crossing over can be easily distributed in the progeny. So with the help of these genetic maps, I can also place, means these genetic maps can help me in assembling the genome, in which I'm having problem in assembling by finding the overlaps. So let's say that I have genome which has, let's say, this thing is sequenced, and here also I have another contig in which this has been sequenced. So now I will go back to the genetic map and I will be sure that, okay, these two fragments will be definitely close to each other, because in the genetic map, which has been very well studied, they were lying close to each other. And in case I've assembled my genome and I want to cross check, there's the possibility that because of certain repeats, I assemble this particular fragment with this particular fragment, because there was some repeat region which was common among the two. So by mistake, what I've done is that I have put together chromosome 1 fragment with chromosome 3. But then I can go back to my genetic map and then I can be sure that what I assembled was wrong. So this is how genetic maps, they can be very useful in giving a hints to a person who is assembling the genome, that what is the right sequence, etc. Obviously, means it will not give the exact sequence, but it can give us a hint of the same and tell us whether we are in the right direction or not. So, are these genetic maps only limited to the names of the genes that we are known? No, there are other maps also that have been generated. There are certain STR maps. So, what are STR maps? They are short tandem repeats. तो जैसे जीन्स के लोकलाइजेशन किया हुआ है ऑन क्रोमोसोम, similarly these tandem repeats, various tandem repeats are there. Now people have also made maps of these tandem repeats, these short tandem repeats on the chromosomes. So if means I can always, similarly to these gene maps, I can also go back to my STR maps and find out that what is the sequence of the various STRs which are present on a chromosome, and that can also give me a hint. Again, similarly, we have SNP maps. So what are SNP? These are short nucleotide polymorphisms. Means at a particular position in the genome, at a particular nucleotide. So maybe if a good amount of percentage of population is showing mutation at that particular location, then we say that it is single nucleotide polymorphism, that at the same place in the genome, some other nucleotide may also be present in a population. So if a particular nucleotide is present in a good percentage of population, more than 1% of the population, then they say that, okay, this is a single nucleotide polymorphism which is present in the whole population. Some population have A at that particular position and some population has T at that particular location. So based on these SNPs also, so SNPs are also randomly divided or distributed throughout the genome. They can be present in any DNA region. It can be in the promoter, in intron, in exon, in untranslated region, anywhere in the genome. And on basis of SNPs also, map has been generated. So you can understand that through these various maps, if you put them together, you will still have a lot of idea that, okay, yellow body comes after DXS 6807, and then white eyes come, then maybe some um, SNP from this comes. So if you put all these together, you definitely have a lot of idea that, okay, these things should be in continuation to each other. So when you have a trouble in assembling the whole genome, and why I'm stressing that there is a problem, because definitely many organisms which have a very big genome size, it is considered a humongous task to actually assemble them, and to be very sure that this is the particular sequence in which this is the particular sequence in that particular genome, that is a very big task assembling. So you have to be helped by multiple strategies to assemble the genome. Now, another map, when I'm talking about genetic map, I should also tell you what are the physical maps. So physical maps is when you place your gene on the chromosomes at the precise location. So that physical map is generated with the help of these fluorescent probes, and this method is called as fluorescence in situ hybridization. So even with the help of physical maps, I can be sure, means it also can somewhere help me in placing my contig, at some place in the genome. So it is generated by methods that directly locate the position of specific sequence on chromosomal DNA. And as in genetic mapping, the loci that are studied can be genes or any DNA region, any DNA marker against which you can generate a probe. Only difference is that in this case, we prefer in current times the fluorescent probe. Okay. So what is done is, because if you want to localize anything on the chromosome, you have to be you should do it at a stage in the cell cycle when the chromosome is most condensed. Okay, so the chromosome, when it is most condensed and has a very beautiful X morphology, it is at the metaphase stage. Okay, if you want to, let's say you generate a fluorescent probe and you do the probing at interphase stage, when the chromosome is more unpacked and it is present in the form of chromatin. You know na, that chromatin, when it folds further, it becomes chromosome. So let's say you go for the unfolded stage and it is in the chromatin stage, and then you do the fluorescent probing, then all these chromosomes will be so interspersed and so spread out with each other. Actually, that means it's said that it's like a Maggie with all these noodle straps with each other. So then it becomes difficult that, okay, this is from which chromosome. So obviously, for better localization, you must go for a stage when it is the metaphase stage in which they are most condensed. And then what you do is that you put your cells on the slide. You treat them with formamide to make these DNA strands, they will open up. I mean, they will be denatured in presence of formamide. And then you add your fluorescent probe and then you do the hybridization. So how will your chromosomes look like? They will look like this. In this case, what they have done is they have used different probes from with different fluorescent dyes. Now thankfully we have multiple fluorescent dyes. We have green, red, yellow, different spectrum of fluorescent dyes which you can use simultaneously. And by generating probes for a particular DNA of interest, you can localize that, okay, this particular gene is present at the end of this particular chromosome, and this gene is present here, this gene is present here. And similarly, you can generate the physical map also by going for the gene sequences that you already know. I mean, at least a part of it you know, and by generating
Ma'am, you are not audible.
yeah, sorry, there was power cut. So am I audible now?
Yes,
Yes,
ma'am.
ma'am.
Yes, ma'am.
Now you
Okay.
are audible.
Yeah, yeah. So means this was the last slide anyway. So is this visible or I have to share it again?
Ma'am, last slide I did not understand, ma'am. Please share it again. Ma'am, the fluorescence part
Yes.
when we are talking about the genes.
So you can see my screen again, right?
Yes, ma'am. Now it is visible.
Huh.
Yes, ma'am. This. Yes, ma'am.
See,
This
this
part.
is, let's say you want to localize any particular gene on the chromosomes. How will you do that? You have to, you cannot see in molecular biology, you cannot see, okay, this gene is placed on this chromosome. You have to find some way to look at it. And one way is that you generate a probe against a particular gene. Let's say you want to go for this self gene, and you want to know that in which chromosome, maybe this is the organism with 13, 15, 20 chromosomes, where it is exactly localized. So what I do is I generate a probe against it. Only thing is that I will label it with fluorescent dye. So then I hybridize it on a cell. I mean, I take that tissue from that particular organism, and then I macerate into single cells, and then I put it on slide. Only thing I should ensure that it is at the metaphase stage. And then I probe it. So I will see that, okay, my this particular gene is hybridizing towards the end of this particular chromosome. We already number the chromosome on the basis of their morphology. This is anyway done, already done for many organisms. And if that is not already done, then you can always number them based on their morphology, and you can figure out that, okay, this particular gene is present here. Then you go for another gene, that, okay, this is present here, this is present here. So in a way, I have physical map, and I'm sure that these three genes, they are close to each other on this particular chromosome. So if while genome assembly by mistake I have put self with axin 1, whatever this gene is, then it means my genome assembly was wrong. It cannot be, because with the help of this genome sorry physical map, I'm sure that self is totally on a different chromosome and axin on a different chromosome. So these two fragments or these two contexts cannot be close to each other. So this is how various these physical maps, these SNP maps, these STR maps, or these gene maps are very useful in genome assembly. They can give a hint that, okay, these two genes or contexts are close to each other. So this is
Ma'am,
all. हाँ बोलो.
ma'am, whenever we are now we in this class we are talking about the genes which are in the close proximity. So, ma'am, we discussed the two methods. So, ma'am, what about the shotgun sequencing apart from clone contig? Because
शॉटगन में
in
तो
the
जो ये है
shotguns
ना, सुनो.
we have
हाँ बोलो.
Ma'am, shotguns maybe we have that identical parts. So what about them? You said
देखो
that it's not.
शॉटगन में ये होगा, हाँ सुनो, शॉटगन में ये होगा, लेट्स से ये कोई ऑर्गेनिज्म है जिसमें 4 क्रोमोसोम्स हैं, ठीक है. Now in shotgun what I do is I isolate total DNA, I break them into small pieces and I do parallel sequencing. अब मेरे पास इन सारे क्रोमोसोम्स का एक साथ मतलब बहुत सारे फ्रैगमेंट्स में सीक्वेंस आ गया है एंड आई हैव टू नाउ असेंबल विद ईच अदर. सो यू कैन अंडरस्टैंड दैट अगर यहाँ पे एक रिपीट है और एक यहाँ पे रिपीट है, आई विल फाइंड इट वेरी वेरी डिफिकल्ट टू असेंबल दीज टू टुगेदर. मेरे को समझ ही नहीं आएगा कि ये रिपीट यहाँ का मैं इससे मैच करूं या ये इससे मैच करूं या इससे मैच करूं. आई हैव सो मेनी डिफरेंट पॉसिबिलिटीज. क्लोन कॉन्टेग में क्या होगा कि मैं ऑलरेडी एक कॉन्टेग ये बना लिया विच कंसिस्ट ऑफ दिस ओवरलैपिंग फ्रैगमेंट्स. एक कॉन्टेग मैंने ये बना लिया विच हैज दीज ओवरलैपिंग फ्रैगमेंट्स. एक कॉन्टेग मैंने ये बना लिया, एक कॉन्टेग ये बना लिया, एक ये बना लिया, बाय लुकिंग फॉर ओवरलैपिंग फ्रैगमेंट्स. अब क्या होगा कि इस कॉन्टेग को मैंने ब्रेक किया और इसकी पैरेलल सीक्वेंसिंग की, इसमें मेरे रिपीट्स आए, लेकिन मेरे लिए आसानी बहुत ज्यादा है इस केस में बिकॉज आई एम डैम श्योर दैट दिस रिपीट I don't have to look for its over, means I have much less trouble in assembling this together as compared to अगर मेरे को ये सोचना होता है ये रिपीट इससे भी मैच करेगा, then I would in shotgun I have so many different options to assemble it. I mean, I can do it wrong. But in case of contact, now I'm limited. I'm very sure that only these sequences have to be assembled. So it becomes much more easier, basically. That's what I want to say.
Yes, ma'am.
ठीक है. Any other question?
Ma'am,
Hmm?
so for this, I mean, for I mean, assessing that our chromosomes are in metaphase, stays in metaphase state only.
Hmm.
So, do we use
How will you do
mitotic
that, right?
poison?
Hmm.
Yes, ma'am. So, do we use mitotic poisons like colchicine and everything? And
Right,
if we
right,
are using
exactly.
them,
Exactly. You have to treat your cells previously with some chemical. It can be colchicine, or it can be there are certain drugs, nocotazole, and so many, in which you can arrest your cells at a particular phase, and then you can let it go for cell cycle, and then you know that okay, iske 10 घंटे बाद it will enter into metaphase. So then you can harvest it. I'm saying ki apart from colchicine also there are other chemicals in the market,
okay,
but
ma'am.
you
So,
have to obviously treat them.
won't they interact? I mean, won't they interfere with the hybridization interaction?
No, no, not at
I mean,
all. No.
okay,
तुम ये सोचो ना कि तुम्हारा,
thank you.
no, they don't interfere. They don't.
Okay, ma'am.
Only thing is that when your cells are on the slide, you have to denature them. Usually it's done with the help of formamide. I'm also not updated with what's other recent methods, but I know that in general, the idea is that you have to make these single stranded so that your probes can bind to it. And probes have to be generated good so that they don't bind to non-specific sequences, and also that they should be fluorescently labeled. So all these techniques, they need optimization for your particular thing. But now there are some standard methods that have been optimized, at least for certain organisms, how to do this fish. Okay, so any more questions?